Hello lovelies. In this video, the brilliant Dr. Uploads is going to go through cloning for your A-level biology. Now, this is absolutely fascinating topic. So we're going to go through the history of it, how we use it at the moment, how we can use it in the future, and then we're going to look at the controversies, the ethics and the morals of it as well. This is something that really easily lends it to a 4, a 6, a longer question. So if you'll make sure you've got all the facts sorted, then over my website, Dr. Rob has thrown a whole set of questions just for you to make sure you understand everything.
Natural and artificial cloning. Clones are genetically identical organisms. Clones form naturally through asexual reproduction, so ones that we've looked at like binary fission in bacteria, but they can also form when an embryo splits into two in the uterus. This forms identical twins. Humans have used artificial cloning methods for many years to create many identical copies of plants, for example, with desired characteristics, or to clone genetically modified organisms. Natural and artificial cloning. Natural cloning. In animals, identical twins form from embryo splitting and asexual reproduction in some insects like aphids. So these are the only two examples of natural cloning in animals, when we get an identical twin from an embryo splitting before it then goes on to develop into two separate embryos, but they are identical, and also some insects are able to asexually reproduce. In plants, natural cloning is known as vegetative propagation. It's the production of plant clones from non-reproductive tissues. There are a couple of methods. Plants can use stolons, sometimes also called runners, like strawberries. They can use bulbs, like onions, or tubers, such as potatoes. They can also, some can clone from their leaf. So these are little plantlet clones on the leaf margins that will fall off, root, and become new plants. There's some other methods, such as rhizomes, suckers, and corms. We can take advantage of plants' natural ability to clone and use cuttings to clone plants. What we're doing is inducing vegetative propagation to get natural clones of a parent plant. You can also use grafting or layering instead of a cutting. So grafting is where you take sort of the stem of one plant and graft it or attach it to the rootstock of another plant. And layering is where we will push stems into the ground in order to try and get them to form runners or stolons. If we're taking a cutting though, we can use any part of the plant. We can use the stem, the leaf, or the root. Here's a quick method for how you could do a cutting from a stem. You need to take 5 to 10 centimetre section of stem from the tip of a shoot, and ideally cut just below a leaf node. That's just the branch where the leaf comes out, as that is where there'll be some meristem tissue, some stem cells that are then going to be able to help to turn those cells into root cells. You need to use sterile scissors or a scalpel or secateurs, and they should be clean and sterile because we don't want to introduce any pathogens to the cut site. You're then going to remove any lower down leaves, dip the cut end into the rooting powder, which will contain some auxins and cytokinins, and then you're going to plant in a suitable soil. So you're just going to remove lower leaves, make sure you've got a couple of leaves at the top so they can obviously carry out photosynthesis, dip the end in rooting powder, and then plant in a suitable growth medium, plenty of water to get it to start, and then you should have a new plant. Very shortly, once it has grown roots enough, you are able to take it out and move it and plant it wherever you want. And it will obviously be identical to the parent plant that you cut it off. You can use a propagator or you can put some clear plastic over it. This is to just keep it warm, increase the humidity, make sure that it's got enough light, high temperature to make sure that it's going to get its best start to start growing roots. So in this case, you remove like a whole branch or a small stem from a larger plant. Then you remove the lower leaves, and the cut end is dipped into rooting powder, which contains growth hormones. Then you put each of these cuttings into damp compost, and the roots begin to grow and a new plant will form. You can also cover them in plastic, like a plastic bag, in order to try and increase and improve the growth. It keeps them warm, it keeps air around them humid, and it helps to encourage root growth. We can also clone plants artificially using the process of tissue culture or micropropagation. So we're going to remove a piece of tissue from our plant, so a small piece. It normally includes a bit of stem because, as we said, there's meristem tissue there, and we use that using sterile forceps. We now have what's called an explant, and we're going to sterilise this in alcohol or bleach because we don't want any bacteria or microorganism growth on the outside of our explant. We're then going to place these explants on a nutrient agar in a petri dish. It's been sterilised again to avoid contamination, and it's going to contain not only nutrients but also some growth hormones. The cells of the explant begin to divide by mitosis and form what we call a callus of totipotent cells, so stem cells. Once that is formed, you can then break this small ball of cells up, the callus up, into smaller clumps of cells, which you can then spread out on your agar plate or a new agar plate. And these will all develop into small, tiny plantlets, and they will all be clones of the first initial plant. We need to be able to talk about the advantages of artificial plant cloning. It allows plant reproduction to occur in any season, so it's not dependent on the temperature or the weather outside. It could save an endangered species of plants, so some orchids, for example, if there's very few of them in the wild, we can quickly make a large number of the same species. Although they're clones, it does mean that the species isn't going to go extinct. It is able to produce a lot of plants faster than if you were going to grow them from seeds, and less space is needed to grow them from seed. So think about how small all those little plants are on a plate. Instead of having to have each individual seed in a big tray with soil, it takes up less space and it is much faster. You're always going to have your desirable characteristics passed on. So if you have a plant that you want to clone because it has specific desired characteristics, if you were to normally or naturally breed that plant, there isn't a guarantee that all of the offspring will have the desired characteristics that you want, because you can't guarantee those genes will be passed on with sexual reproduction, whereas you can if you're going to clone your plant. The disadvantages of this method of cloning: the production costs are very high, much higher than just growing by seed. There's a contamination risk that all the plants could be killed if they get infected with some kind of disease, especially during the process if sterile techniques are not used. The cloned populations also have no genetic variability, so they could all be wiped out by another disease. Again, although you can control the desirable characteristics that are passed on, also any undesirable characteristics will always be passed on, and you cannot stop this. Artificial animal cloning. One method of animal cloning involves embryo transplants. This is where an embryo is split into individual cells. So a fertilized embryo starts to divide and produce new cells. Each of them are identical because it's happened through mitosis. So once that starts to happen, we can take individual cells and then transplant them each into a different host animal. In this example, we would get 4 identical piglets, but they would be born from 4 different pigs. Okay, so this is a way of producing identical organisms, clones of each other, but using different host mothers. So they can be born all at the same time. Another type is somatic cell nuclear transfer. This will use 3 separate individuals. So I have cow A, cow B, and cow C. So I remove a body cell from cow A. This is the animal I want to clone. And I remove the nucleus from this body cell. Cow B, I take an egg cell out and I remove the nucleus from that egg cell. It's called enucleation, or an enucleated egg cell. Then I'm going to take my nucleus from my body cell and I'm going to fuse it into the enucleated egg cell. We do this by stimulating it with electricity called electroporation, and then it stimulates to divide as well. So that electric shock helps to fuse the two together and also stimulates it to start dividing into an embryo. Then once the embryo has started to divide, so we know that it's healthy and it's going to continue growing, you're going to place that into the surrogate mother, which is cow C. And then this should proceed like a normal pregnancy in order for the animal to be born. So the calf born from mother C will be a clone that is genetically identical to the animal A, because we took the nucleus with its DNA in and put it into the egg cell. So the DNA from cow A will be in the nucleus of every single cell in the organism that's born from cow C. Again, both of these types of cloning are illegal in humans. So just like with genetic modification, cloning, especially animal cloning, is not allowed in humans. So there is also non-reproductive cloning, so not actually producing a new organism. The main form of this is therapeutic cloning. This is where cells are cloned in order to grow replacement cells or tissues for patients. So it could be, for example, skin cells grown in vitro in a lab in order to create a skin graft to treat a burn. Growing islet cells for a pancreas for someone who is type 1 diabetic, nervous tissue for spinal injuries as well. And this is kind of a type of cloning because we'd be taking genetic information in the nucleus or a cell from the patient and then making sure we multiply that cell multiple times, growing that cell in vitro outside of the body and then replacing it. So it would be cloning or using the patient's own cells, which is important because it reduces the chance of rejection. So it's a way of growing cells or tissues that can be transplanted that are not going to be too different and are not going to cause the immune system of the person to react. Also, embryos can be cloned to be used for scientific research. So not cloning embryos in order to make multiple new organisms, but cloning embryos so that we can look at that stage of development. So that stage from transitioning from an embryo and how the process of differentiation starts, and being able to use that for genetic analysis to look at how genetics actually controls that initial process of development, and then also give us an opportunity to maybe test drugs or medicines on human cells that are dividing from an actual embryo. Or to use the stem cells in embryos to grow tissues in order to test new drugs or medicines. We need to be able to discuss both the advantages and disadvantages of animal cloning. We can breed from infertile animals, so animals that have desired characteristics that you want but wouldn't otherwise be able to breed for whatever reason. We can take a body cell from that organism and clone it in this way. Similar to plant cloning, desired characteristics are always going to be passed on. It can also help in save endangered species. And the other useful technique about this is that we can use it to produce stem cells for disease research, or produce embryonic stem cells that have the DNA of the patient that we want to do something with stem cell treatment or stem cell therapy on. Also, we can reproduce lots of genetically modified organisms. So once we've made a genetically modified organism, we can clone that organism rather than having to remake a load of genetically modified organisms. It's very difficult and expensive, and there is a research to suggest that clones may have a shorter lifespan, which then brings up ethical issues about whether it's right or not to make cloned organisms. Again, similar to plant cloning, they have low genetic variability, so a population of cloned organisms could die because they're going to be susceptible to diseases. Also, there is an ethical issues surrounding the production of embryos for stem cell use, so producing these embryos, using them for stem cell research and then maybe destroying them or not using them or taking them further is seen as destroying a life. So sometimes there can be objections around producing embryos for disease research rather than to produce organisms.
Ouch! This is why in some videos I explain scratches.